The first antigen is a neoantigen. As the name shows, it's a neo indeed antigen. It's the product of mutations. For example, you can see here we have a normal cell, and this is an example of a normal protein with different epitopes. You know all of them. A protein, a big antigen with several epitopes. They are all normal epitopes, and they are expressed on the, what is this? What is this?
MHC1.
MHC class 1. Why is it MHC class 1? It's a normal nucleated cell, it's not an APC. For example, it's a liver cell. On the APCs we have MHC class 2. MHC class 1 is in all normal cells. So this is a normal cell that it has MHC and normal epitopes. Okay? When the epitopes are normal, we won't have any immune response. We won't have any response from T cells because everything is normal. We have tolerance against normal antigens and their epitopes. In the second one, we have a mutation on the DNA, and as a result of mutation, now we have a new epitope. Immune system is very sensitive to any changes in normal proteins of the body or normal macromolecules of the body. Immune system is very sensitive. Any tiny changes can activate immune system. So we have a mutation and a change in the normal protein that is not normal anymore. We have new epitope. The rest of the protein is normal. Just this part as a new epitope is abnormal. So that's new antigen. We have the new antigen on the MHC class 1, and then we will have T cell activation and immune response. Okay? So this is one way that immune system can discriminate between normal and abnormal cell. By this discrimination that is being done in our body regularly, all the time. Immune system is detecting, for example, those kind of antigens and kill the target cell. Another antigen is antigens of oncogenic viruses. The viruses that can induce cancer, can cause cancer, HPV, HCV, HTLV, HIV, and other viruses. So here we have, you know that when we have a virus gets into the cell in order to use the translation and transcription machinery of the cell to proliferate, and some of the antigens of the, this is the protein of the virus, and some antigens of the virus are presented by the MHC class 1 of the cell. So they are the viral antigens. And viral antigens are foreign to immune system. Again, they are foreign, they are not normal, they are not self-antigen, so we will have activation of immune system. Another example, the first two was their tumor-specific antigen. This one is tumor-associated antigen. Overexpressed cellular proteins. As the name shows, they are overexpressed. We have higher production or unwanted production of them. They are normally silenced in normal cell, or we have a very low level of their production. And sometimes we have excessive amount of those antigens. For example, there are different types of overexpressed cellular proteins, including cancer testis antigens. They are the antigens that are normally expressed in gametes and trophoblasts, but not other cells. We have some genes that they are normally expressed in these two types of cells. But as a result of mutation in the cancer, we will have their expression in some cancers. They are called cancer testis antigens. Hundreds of cancer testis antigens have been identified until now, and they are used as a therapeutic target or tumor marker in the laboratory. For example, melanoma-associated antigen it's an example of cancer testis antigen. You can see here, for example, in the normal cell, as a result of methylation or any other biological changes, we have no expression, for example, of these antigens in normal cell. But as a result of mutation, we have activation of their expression, unwanted expression of that antigen in the cancer cell, and these antigens are presented by HLA, and it can activate immune system sometimes, not always. And hundreds of them have been identified until now. Another overexpressed cellular proteins are those that, as a result of mutation again, we have abnormally a very high level production of them in the tumor cell. For example, HER2 is a good example of tumor marker or tumor antigen. HER2 is a growth factor receptor. HER2 is a name for growth factor receptor. As the name shows, it's a receptor for growth factor. Most of the normal cells have HER2. Most of the normal cells have HER2. But in cancerous cell, we have abnormal high level production of the HER2. For example, if a normal cell have, for example, 10 HER2, then the numbers are just examples, they are not scientific. 10 HER2, in the cancer cells, we have thousands of HER2. And you know that, so that the number of the HER2, the level of the HER2 is abnormally increased in cancer. This one is normal. And you know that increasing the dose of antigens could change the immunogenicity of the antigen. By increasing the amount of antigen, the immunogenicity changes. So here we have a very low level of expression. For example, it's not immunogen over there, but here we have a very high level of its expression. So now it would be, or it might be, immunogenic. And HER2 is used widely in several types of cancer, including gastric cancer, ovarian cancer, breast cancer, both for a therapeutic immunotherapy, as an immunotherapy target, as a detection marker, as a diagnostic prognostic marker. For example, you can see here that we have a normal expression, for example, 3 antigens here, but as a result of a mutation, we have a large amount of that protein. So we have higher expression of the antigen. And this higher expression can be detected by immune system or in the laboratory. Some overexpressed cellular proteins are tissue-specific. Differentiation antigens are tissue-specific antigens. I've talked about them. The antigens that are specific to each tissue, for example, kidney antigens, lung antigens. Some cells are just lung cells, and there are some antigens that are only expressed in lung. For example, you know that CD3 is only expressed on CD3 on T lymphocytes, yes. CD4 on T helpers. So they are indeed cell-specific antigens. We have such antigens in the lung, kidney, and few other tissues as well. So they are called differentiation antigens or tissue-specific antigens. For example, this enzyme is a differentiation antigen in melanoma, skin cancer, this type of skin cancer. Another type of antigen is oncofetal antigen. Oncofetal antigen. Onco is from oncology and it's from fetus. They are proteins that are normally expressed in the fetus, and they have normal physiological role in the fetus, in the baby before birth. Okay? But after birth, their production is stopped normally. After birth, we do not have their expression. But in some cancers, As a result of mutation, we have again unwanted expression of them. So they are antigens that normally are in the fetus, but in some cancers and some inflammatory conditions, we have their expression. So they are called oncofetal antigens. Some oncofetal antigens now are detected in the laboratory. For example, CEA, carcinoembryonic antigen, which is named CD60C, is normally expressed in gut, pancreas, and liver in the first 2 trimesters of gestation in the fetus. Gestation means the period of pregnancy, the time that the fetus or the baby haven't given birth. Before birth, it is called gestation. In many carcinoma, including colon carcinoma, pancreas carcinoma, stomach carcinoma, and so on, we have expression of CEA. And for example, AFP, alpha-fetoprotein, is another oncofetal antigen, which is normally expressed in yolk sac and liver of fetal life. Fetal life is another name for gestational age. So they are expressed, it is expressed normally in yolk sac and liver of fetal life, but in some cancers, including hepatocellular carcinoma, germ cell tumors, and some others, we have the re-expression of that antigen. So it's again tumor-associated antigen, abnormal expression in terms of time now, not the level. The previous one was abnormal expression here too, was an example of abnormal expression in terms of the level of the protein. Here we have abnormal expression in terms of the time. They are normal for fetal life, gestation, but after birth, they normally we do not have their expression, but in some cancers. Another antigen is altered glycolipid or glycoprotein. What is a glycolipid? What is the difference between glycolipid and lipid, or glycoprotein and protein? I've asked it once before. Yes, protein, which is bound to carbohydrate, which is said glycoprotein. Lipid, which is bound to carbohydrate, which is called glycolipid. Okay? They are the topics of biochemistry. We have lipid and glycolipid, protein and glycoprotein. So, in some cancers, in normal cells, we have expression of some glycoproteins and glycolipids on the normal cells. Let me check. I have a good figure here. For example, this is a protein. This is a protein chain, and in normal situation, there are some carbohydrate on it with this form, for example, and with that number. The red one is protein, the blue ones are carbohydrates, both in terms of their number, for example, just 5 of them, and with that shape. Sometimes, as a result of cancer, we have a protein again, but the shape of the glycoprotein will changes, for example, in this way. So we have a changed glycoprotein. The type of the carbohydrate that is bound to this protein is changed here. And these changes, I've told you, any changes can activate immune system, can sensitize immune system. Or in another way, we have another example, that the shape isn't changed, but the number has changed. We have higher number of that protein, that glycosylation. The process of adding carbohydrate to protein is called glycolization. So here the pattern of glycosylation is changed. Here we have another type of glycosylation changes. Here the shape of indeed the type of carbohydrate is changed. Here the number of the carbohydrate is increased. Both of them are changes. An immune system can detect both of those changes. Clear or not? Do you know what is altered glycoprotein or glycolipid or not? Guys, please. Yes? Yes? Ladies? Of course. That's why I'm asking you. So, this one is a normal. That one is a normal. Please have a look here. Is a normal glycoprotein as an example of glycoprotein or glycolipid. This is a normal glycoprotein. And this red one is the protein part of the glycoprotein. And the blue ones are carbohydrates. Okay? We have a protein that some carbohydrates are bound to that protein. For example, glucose, mannose, any carbohydrate. But in some tumors, the pattern of the binding of those carbohydrates to protein is called glycosylation. And it is done by some enzymes. The binding of those carbohydrates to the protein is made by some enzymes. In some proteins, the pattern of glycosylation changes. For example, here we have that protein. The protein is normal. But the carbohydrates are changed. For example, instead of glucose, we have saccharose. Instead of glucose, we have mannose. This is a different carbohydrate. The type of the carbohydrate bound to that protein is changed. So we have a new glycoprotein or altered, changed glycoprotein. And that changes can activate immune system. The number is same. Here we have 5 of them, and here we have 5 of them. In another example, the type of the carbohydrate hasn't changed, but the number of glycosylation, the amount of glycosylation has changed. For example, we have just 5 carbohydrates bound to the protein. Here we have, for example, 10 of them or 9 of them that are bound to carbohydrates. The number is increased. And again, we have changes. And these changes can activate the immune system again. Okay? We are talking about one type of tumor antigen which is named altered glycolipid or glycoprotein. Here is an example of glycoprotein. And there are some glycoproteins, for example, MUC1, mucin1 is an altered glycoprotein which is now is being used in the research and clinical as a target for the detection and treatment of some cancers. No question? No comment? So until now,
why it's expressed on the normal cell?
because the normal cell lives for that. They are the, for example, normal part of that cell. The cells, all of the cells, most human cells have glycoproteins, and most molecules on the surface of cells are needed for their normal biology. And we have so many proteins, glycoproteins, glycolipids on the cell membrane of all cells almost. They are needed for their normal life. No question? So, until now, I summarized you the antigens of the tumor. Indeed, now you know that how immune system can detect cancer, how immune system can discriminate between cancer and normal cell. You know the answer of this question. Am I right? By detection of those antigens. Oncofetal antigens, altered glycoproteins, neoantigens, viral antigens, all of those antigens, you know them now, are the way by which immune system can detect, can discriminate, can differentiate between normal and cancer cells. And now, after detection, immune system is going to do something to kill the tumor cells. We are going to talk how immune system is going to kill the tumor cells. How immune system is going to deal with, to respond to tumor antigens or tumor cells after detection of the antigens. What do you think? How immune system can kill the tumor cells?
By cytotoxic T cell,
By cytotoxic T cells. Why do you think so?
because it is have a CD8. CD8.
Cytotoxic T cells are CD8, yes,
Yes,
positive.
bind to the MHC1 that express our self antigen, and the cytotoxic T cell is for virus antigen.
Intracellular antigens.
Yes,
Intracellular antigens. Because cancer, the antigens of the cancers are intracellular antigens. So as the cancer have intracellular antigens that are expressed by MHC class 1, so the main part of immune system that can recognize and can kill the cancer cell is CD8 positive or CTLs or cytotoxic T cells. What else? What other cells are involved? So the first cell is CTL or CD8 positive cells.
Maybe macrophages.
Macrophages. What macrophage can do against cancer? Phagocytosis. What else? The role of macrophage first. So we need when we have CTL, we need APC, macrophage. Can you tell me another important APC for activation of naive T cells? Which APC? Not, which one? No. APC. Ya Allah, APC. Dendritic cell. Dendritic cell was the most efficient APC for activation of naive T cells. So we have dendritic cell and macrophage that they express the antigen for activation of naive T cell. What other cells are involved? NK cells. NK cells can directly kill the tumor cells. I told you that NK cell is very important in from innate immune cells against tumor or intracellular antigens, tumor and viral infected cells. What else? Which one? Yes, T helpers. Which T helper? CD? No. CD? CD8, CTL, yes, I've written here. We are talking T helper. It's a CD4 positive. T helpers are CD4 positive. So we had T helper 1, 2, and so on. Which T helper is involved here more effectively? One or two? Which one?
Two.
1 or 2 or 17? Which one?
Two.
Interferon gamma. What else? I've told you that TLP1 is important to provide help for both APC and CTL in cellular immunity. We had 3 cells in cellular immunity. APC is mainly dendritic cell, CTL and TLP1. They are the main cells of cellular immunity. Do you remember? But NK cells is in innate immunity. And those cells are from innate as well. But those one are from adaptive immunity. Those are the main. When you want to talk about the immune response against anything, first of all, you should talk about the first layer of immune responses. First layer of immune response is always innate immune responses. So in innate immunity, we have NK cells that is very efficient, is very important against tumor, and APCs that they activate adaptive immunity. And in adaptive immunity, we have 2 arms, humoral and cellular. Which one is important here? Cellular, because the antigens are.
Intracellular.
But does it mean that we do not have humoral immune response? We have. I've told you that always we have both of them, but the other one is more effective, more protective. We have humoral immune response. We have antibody against cancer, but it's not effective or as effective as a cellular immune response, cellular components. Clear or not? So you know all of them. From now on, you know almost all of them. First of all, innate immunity, we have some component in immunity, including neutrophils, eosinophils, NK cell, macrophages, dendritic cells, you know all of them, and their functions, and complement system. Okay? And then adaptive immunity, B cells, T helpers, cytotoxic T cells. They are all the things we have in immune system. So, the CD8 positive CTLs or cytotoxic T cells are the main cells, the most important cell against the cancer. I've written here because I've written here the first cell because it's the most important cell, the most effective cell. That could, how CTL? So now we know that CTL is important against tumor. How CTL become activated? How can CTL, if you want to tell the process of CTL activation, what do we need for CTL activation?
Presenting antigen. Presenting antigen.
Presentation of antigen by APCs. Presentation of antigen, detection of antigen. So the detection of antigen is by APCs, MHC class 1, and what else? Co-stimulators, including B7 and MHC class 1, because it's a CTL. It needs MHC class 1. So we will have here, what is this? This one, no. Receptor, the name of receptor. No, this one is MHC. Cytotoxic CTL. So now we have other cells that help CTL, or you will see in the next slide, you know. Now we have activated CTL. How CTL can kill the tumor cell? Please wait, sorry. How a CTL can kill the tumor cell? Wait. The rest of the students, please. Guys, ladies, gentlemen, Hajis, I've repeated those things several times. How a CTL can kill the tumor cell? What is what kind of tools a CTL has to kill the tumor cell? Does it use a gun or a knife?
It has two mechanisms. The first one, like macrophage, natural killer cells, the same CDA cytotoxic.
Yes.
And the second one, the basal and basal.
Two mechanisms, yeah, fast ligand and fast. Yes. The first one is the mechanism similar to NK. Can we explain it a little more?
It's like two
enzymes?
Two enzymes.
Or two proteins?
Two proteins, granzyme,
Granzyme,
and porphyrin.
CTLs have granules, and inside the granules there are two important proteins, perforin and granzyme. Perforin makes hole on the tumor cell. For example, if this is tumor cell, perforin makes holes on the tumor cell, and granzyme go inside the tumor and induce apoptosis. And another mechanism is binding of fast ligand and fast of the CTL to the target cell. So again, APC and APC it is presenting tumor antigen by MHC class 1 to CD8 and co-stimulator, and for and by MHC class 2, you know that APC has both MHC class 1 and 2. You know this. And APCs are the only cells that have co-stimulator. Why do we need co-stimulator here? What is the role of co-stimulator? If there is no co-stimulator, what will happen? Energy, yes. Because for naive cells, we need co-stimulator. We need two signals. As a reminder. So, two signals for CD8 and of course two signals for CD4. Which one of CD4 we have here? T helper. T helper.
1.
1. It will produce cytokines, and of course it will produce. I think I have another slide for this. CD40 and CD40 ligand. Do you remember? CD40 ligand and CD40. So T helper will help APC and CD8 simultaneously. It's a helper T cell. By the help of T helper, we will have increased activation and proliferation of the CTL. So, as the recognition of antigen, the next step is after activation. The first step after activation, proliferation, clonal expansion, and differentiation to a effector cells and memory. Effector and memory cells. So the first step is detection of antigen, and then activation, and then clonal expansion and proliferation and differentiation to CTL. Now you can see here that there is no granule here. The naive CTL cannot kill the tumor cell because it doesn't have granule. But here you can see many granules inside it. That it has perforin and Bronson. So you can see here the crosstalk between TLR1 and APC, CD40 and CD40 ligand. So what is the role of TLR1 in immune response against cancer? What is the role of TLR1 in immune response against cancer? Production of cytokines, yes. What else? Production of cytokine, indeed helping for CD8 positive cells, and expression of CD40 ligand to help APC. Okay? It's a T helper. How it can help other cells? By helping to CD8 by production of cytokine, and by helping to APC by expression of CD40 ligand, and of course some cytokines. Okay? The T helper 1 use two ways to help the cellular immunity: production of cytokine and expression of CD40 ligand. CD40 and CD40 ligand. Okay, that's for cellular immunity. But I've told you that we have antibody against immune system, antibodies. But the role of the protective role of antibody is not as strong as cellular immunity. They are not protective so much, but we have antibody. So if we have antibody against tumor, antibody is produced by B cells. So we need B cell, we have B cell involvement as well. So we have other T helpers involvement as well. So how an antibody can fight with cancer? We have a cancer cell and antibody. How an antibody can fight with cancer?
By activate the complement system.
By activation of complement. When we say antibody, you should think about the functions of antibody. Activation of complement, opsonization, ADCC. Those are the mechanisms that antibody can activate NK cell, can activate complement, and can activate by opsonization phagocytosis. They are the role of antibody. You know the role of antibody, the roles of antibody, the functions of antibody. You should think about it and find the functions and find the relationships. NK cells. NK cells use three mechanisms I've told you. In three different ways, an NK cell can become activated. The first one is MHC class 1 downregulation. I've told you before. Some cancers that I'll talk about it downregulate or prevent MHC class 1 expression in order to escape from immune system. If a cancer cell doesn't have MHC class 1, what will happen to that cancer cell? Think about the cancer cell that doesn't express MHC class 1 anymore. What is going to happen to immune response against that cancer?
The cell cannot recognize
CTLs cannot recognize that and kill. So the recognition of tumor cell is by MHC class 1. So when there is no MHC class 1, there is no recognition by CTL. Another way, so the cancer downregulate the MHC class 1 in order to escape from CTL killing. But immune system, I told you that there is competition between tumor and immune system. But immune system has another cell to fight with the cells that do not have MHC class 1. Downregulation of MHC class 1 will activate NK cells. And expression of some stress receptors. Do you remember? Stress proteins. I've told you that stress proteins are some abnormal proteins that are expressed on abnormal cells only, not normal cells. And those stress proteins, including those one, can activate NK cells. NK cells have a receptor for those stress proteins. Another way is ADCC. You know ADCC. Antibody against tumor cell, binding of antibody to tumor cell, and then binding of NK cells with its receptor. What is this receptor? This receptor on the NK cell, which binds to antibody. FC receptor. By binding of FC receptor on the NK cell to antibody, we will have activation on NK cell. Again, NK cell has granule, and inside the granule we have perforin and granzyme. And again, after releasing of the content of granule on the tumor cell, we will have apoptosis of tumors. Macrophages. It's important to say to you that we have two types of macrophages. Macrophages M1 and M2. M1 macrophages are inflammatory macrophages. But M2 macrophages are anti-inflammatory. They are indeed a regulatory, a suppressory macrophage. But this one is activatory macrophage. Like regulatory T cells and T helpers. Regulatory T cells are suppressory cells, but T helpers and other cells are activatory. So we have two macrophages, M1 and M2. So M1 macrophages are in favor of the patient. They fight with the cancer by phagocytosis, by production of cytokines, you know them. But M2 macrophages are in favor of tumor. Tumor uses M2 macrophage to escape from immune system. I'll talk about it. Use M2 macrophages to prevent activation of other cells. I'll talk about it in the next slide. And M1 macrophages are called classical macrophage as well. You need to know this. M1 macrophage, the other names for M1 macrophage, classical macrophage, or inflammatory macrophages. And M2 macrophage sometimes are called alternative macrophage or anti-inflammatory macrophages. How macrophage can recognize the T cell, the tumor cell? By PRR. PRR bind to what? PAMs. Here we do not have DAMPs, damage-associated molecular patterns, that they activate macrophage. If we have M1, we will have protective immune response. If we have M2, they are in favor of tumor. And macrophage will produce interferon gamma that can increase the activation of TFR1, CTL, and NK cells. So by production of interferon gamma will help to those cells that all of them are very important against tumor cells. Let me skip this video. That's enough for now. Let me check the time. Yes, you have 15 minutes to rest. I'll get back to class in 10 past 10. Okay?